Hi everybody, today we're going to be going over the Unit 5 exam review. So your first task is to look at this diagram and label it. So let's go ahead and do that together. And so this right here is, oops, it's not writing. Let's make sure everything's okay. There we go. All right, so this is the chromosome right here. Okay, and this is all happening in the nucleus. So just to get our bearings where we are, this is the DNA. All right, and this unwound part is the gene. Okay, so remember we only copy one gene during transcription. And the enzyme that's going to get this done is this right here. It's called RNA polymerase. Okay. And this is our mRNA copy right here. Okay, so that's that part, nothing too fancy there, but this one gets quite a bit more complicated. And so let's start over here with the nucleus, because that's where transcription happens. And so that's where the mRNA is made here. Okay, so that's one mRNA coming out. This one is already out and being translated. So 3 is pointing to mRNA as well. Okay, and let's see here. So this is a ribosome right here. So is this. Okay, and these 3-letter codes are codons. And I believe that's what number 5 is referring to right here, is codons. And this right here is the tRNA, so 7 is the tRNA. tRNA is attached to an amino acid. And this right here is our codon again, or I'm sorry, anticodon. Okay, so the anticodon should be complementary in sequence to the mRNA codon. And all right, so these tRNAs are landing right here, and we're going to start to make peptide bonds between the amino acids. All right, and as the amino acids attach, the unloaded tRNA will move out to go pick up more amino acids. So these are free-floating amino acids. All right, and those come from your food. So any free-floating amino acids that came from your food are going to eventually stick to a tRNA and get used to make proteins here. Now, what I want you guys to do on your own is to look at your codon chart in your notebook and make sure that you look up what these codons are so that you can name the proper amino acid that would come and land. So I'd like you guys to write in the 3-letter code here, here, and here at least. If you guys want to look a little bit down the line here, you could probably figure out the other ones as well. But that's not going to be required, okay? But definitely tell me what these 3 are. With this one, you have to be careful because you can't take that ACA, that's the anticodon. You got to figure out what the codon would be, all right? So that your chart is based on the mRNA strand. Okay, let's move on. All right, proto-oncogenes versus tumor suppressors. So tumor suppressors stop cell division. And they could lead to apoptosis. Remember, that is when the cell programs itself to die. Proto-oncogenes, these produce proteins that promote cell division. And when they're mutated, could turn into oncogenes and cause cancer. Okay, both could promote cancer if they are not working correctly. All right, so they turn into oncogenes. That's the mutated form. Okay, so this question was right out of your notes. Okay, so this next part is referring to this diagram. So which cell's in anaphase? Looks like E is. So anaphase is where the sister chromatids pull apart. A and F. All right, so you see both of those kind of, well, they look similar. They have the nucleus still either intact or almost all the way intact. And the chromosomes aren't lined up in any particular way, so those must be prophase. Okay, so this is probably early prophase because the nucleus is still pretty much all there. This is later in prophase because pretty soon, once we get to metaphase, there's no nucleus at all. And structure X, that is the, you could have put centrosome, or we'd accept centrioles. All right, so centrosome is sort of the centrioles with all the spindle fibers. Structure Y, so those are these fibers, those are the spindle fibers. Kind of almost looks like it's pointing to the nucleus. Which one of these is not in mitosis? So that means which one would be an interphase, and that's D. So it has not started dividing. There's no chromosomes present. It all kind of looks like a uniform color in there. And the main change in B, okay, so in B, it kind of looks like it's already done anaphase, but we start to see the nucleus coming back. And so the nucleus starts to reappear. Okay, and we also see the cleavage furrow here. Okay, and the end product is two identical daughter cells. All right, next one is this chart. So cell growth occurs during interphase. Nuclear division, that's mitosis. Where we distribute chromosomes, that's mitosis. Protein production, that's going to be high when the cell is growing, so that's going to be in interphase. We're going to duplicate the chromosomes during S phase, and that is part of interphase. DNA synthesis is the same thing as duplicating chromosomes, so that's going to happen during interphase. Cytoplasm dividing immediately after mitosis. And mitochondria and other organelles are made during interphase. This is going to be during the G1, G2 times. All right. So somatic and germ cell, how they differ, which one's more detrimental. So only germline mutations get passed down to the next generation. So these are passed down through what we call the sex cells, so that could be either sperm or egg. All right, somatic mutations occur in somatic cells. And somatic cells, they do not donate DNA to the sperm and egg, so they do not get passed down. So somatic cell mutations do not get passed on. All right. So let's take a look at this. So this is the original DNA and the mRNA transcript. So let's go through here. AUG, UGG, AAC, CGC, GA. And so you're going to look at your codon chart and you're going to look up all of these. AUG, of course, is our start codon or methionine. And you're going to want to make sure you get all the correct amino acids there, and you should get to a stop right here. All right, now in this mutated sequence, you see that C has been replaced by T. Okay, and so when you do the mRNA transcript, okay, what's going to end up happening, if we go through all of this, okay, you'll find that this is still methionine here, but this is going to be a stop codon. So we wouldn't actually ever even translate any of the other ones. So what this is a substitution. So we've substituted one letter for another. And so it's not just a one amino acid change, it's a premature stop signal. So that's going to result in a far shorter protein. Okay, so we're going to circle substitution here. And why would it be non-functional? Because it's too short. Too short, and so it wouldn't take on the shape of the normal protein. Oops. Okay, so study hard. This review is only a sampling of what's going to be on the test. You're going to want to make sure you review everything in your composition notebook, everything in the chapters, everything in all the activities that we've done and passed back. Definitely take a look at your old quizzes and see what mistakes you made so you don't make the same mistakes on the test. All right, good luck, and I'll see you guys on Monday.